welcome to the deep dive. Today, we're embarking on a really fascinating journey into carbohydrate metabolism.
Yeah, it's fundamental stuff.
It really is. This isn't just about, breaking down the food on your plate. It's about uncovering the fundamental energy engine, the one that powers every single cell in your body.
Absolutely.
We got a fantastic collection of sources here, really perfect for anyone curious about how our bodies work, especially if you're just starting to explore the incredible complexity of human biochemistry.
That's right. Our mission for this deep dive, you could say, is to extract the absolute essentials, those kind of knowledge nuggets on how your body handles carbohydrates. We'll track them from, that first bite of food all the way down to the tiny, intricate processes of cellular energy production.
Right.
And we'll use this foundational understanding as our, let's call it, metabolic compass. It'll help us navigate and really understand some key clinical conditions later on.
Okay, so if you've ever wondered where your body gets its instant fuel, or maybe why some people just can't tolerate dairy, or even what actually causes that burning sensation in your muscles during an intense workout, well, you are definitely in the right place.
We hope so.
Yeah. We'll even throw in some memorable metaphors, some angers to help things stick, and maybe a couple of questions for you to ponder as we go. We'll circle back to the answers later in our conversation. All right, let's dive in.
Let's do it.
So let's kick things off with the absolute basics. What exactly are carbohydrates? We hear about them constantly, but at a cellular level, what makes them so important as the body's primary quick-access fuel source?
Well, okay, at their core, carbohydrates are essentially chains of carbon, hydrogen, and oxygen. Think of them like the body's quick burn logs for its energy fireplace, maybe.
I like that analogy, quick burn logs.
Yeah. And what's fascinating about their structure is that they contain many hydroxyl groups. You can almost picture these as little chemical hooks that just love to grab onto water molecules.
Oh, okay.
So this makes carbs water-loving or, hydrophilic, which is super important for how they dissolve, how they move around, and how they interact in our very watery cells and bloodstream.
So not just simple sugars then, but these complex structures. And I've heard they're partially oxidized. What's the practical upshot of that for our bodies? Does it make them like a preferred fuel in certain situations compared to, say, fats?
That's a great question. And yes, that partial oxidation is a key design feature, really. It means they require less oxygen for complete breakdown compared to fats.
Ah, less oxygen.
Right. Which makes them a faster, super efficient fuel in situations where oxygen might be a bit limited. Think about sprinting, for example.
Right. Exactly.
And you're right, this carbohydrate family is incredibly diverse. So let's zoom in a bit and see the different forms these energy-packed molecules can take. We should probably start with the very smallest building blocks.
Okay. The basics.
Exactly. We begin with monosaccharides. These are the single sugar units, like you said, the single bricks in our sugar building. They typically have between 3 and 9 carbon atoms.
Single bricks. Got it.
The most important examples for us really are the 6 carbon sugars, the hexoses, things like glucose, fructose, and galactose. Glucose is definitely the star player here.
The main rung.
For sure. And it's a subtle detail, but incredibly important. These sugar molecules, they naturally exist as diisomers. That's the form our body uses. And for those with 5 or more carbons, they also form ring structures.
And these rings, they can form in slightly different orientations, can't they? Like alpha or beta forms. And I heard our enzymes can actually tell the difference. Why does that matter so much?
Oh, it matters hugely. Think of it like a left-handed versus a right-handed glove.
Okay.
Our enzymes are like molecular keys, and they only fit very specific locks. So whether a glucose molecule is in its alpha or beta form dictates whether our body can break it down for energy, like it does with starch, which has alpha bonds. Or if it passes through largely undigested as fiber, like cellulose, which has beta bonds. It's this tiny structural difference that dictates massive metabolic outcomes.
Wow.
Also, keeping tabs on these monosaccharides in the blood is absolutely critical. Glucose concentration, normally around 5 millimolar or 5 millilemp, is vital. Too high, you're looking at potential diabetes. Too low, hypoglycemia.
And fructose or galactose.
Yeah, elevated levels of those can signal specific, often inherited clinical conditions too.
Okay, so those are the single bricks. What comes next?
Moving up just a little in complexity, we have disaccharides. These are simply 2 single sugar units linked together, like snapping 2 Lego bricks together.
Okay, 2 bricks.
Exactly. This connection is called an O-glycosidic bond, and it's formed by a process called condensation. You can imagine a tiny water molecule being released as they join up.
Right.
Common ones you definitely know are lactose, that's the sugar in milk made of galactose and glucose. Then there's sucrose, which is just table sugar, that's fructose and glucose. And maltose, which is simply two glucose units joined together.
So single bricks, double bricks. What about the longer chains, the really big ones?
Right, then we scale up quite dramatically to the polysaccharides. These are the true complex carbohydrates. Think of these not as bricks, but as entire high-rise buildings made of those sugar bricks. Huge structures, hundreds or even thousands of monosaccharides linked together.
Okay.
For energy storage, animals like us store glucose mainly as glycogen. It's a highly branched structure found mostly in our liver and muscles, like a readily accessible stockpile.
Our emergency stash.
Kind of, yeah. Plants use starch for their energy storage, which is composed of amylose and amylopectin. Similar idea, but structured a bit differently. And then there's cellulose. It's a key structural component in plants, gives them rigidity. It's also made of glucose units.
But wait, here's that alpha-beta thing again, right?
Exactly. The bonds in cellulose are beta-1,4 glycosidic bonds. And here's the important detail for us. Humans simply don't have the enzymes needed to break those beta bonds.
So we can't digest it.
Precisely. Which is why cellulose passes through us largely untouched, acting as dietary fiber. It's crucial for gut health, keeps things moving, but we don't get energy from it.
Okay, so we've outlined this huge carbohydrate family, from single units right up to these massive branched structures like glycogen. But, of course, these complex molecules, they don't just magically appear inside our cells ready to be used as fuel. They have to embark on a journey first. How does our body actually break down these big structures and get them from our plate into the bloodstream?
Right, this is stage 1, digestion and absorption. And it's an extracellular process, meaning it happens outside the cells, primarily in the gastrointestinal tract. You can think of it as a multi-stage deconstruction project, taking apart those complex carbohydrate buildings piece by piece.
And that deconstruction actually begins right in your mouth, doesn't it, with salivary amylase?
Exactly. That enzyme starts snipping the amor bonds in its starch and glycogen, breaking them down into smaller fragments like dextrins and some disaccharides.
Just the beginning, though.
Just the start.
Yeah.
Then what? Then, as the food moves down into the small intestine, pancreatic amylase jumps in and continues breaking those A1,4 bonds. But the real heavy lifting, the final breakdown to single units, happens right at the surface of the small intestine, at the brush border.
The brush border, okay.
Yeah, this lining is equipped with specialized enzymes called disaccharidases. They're like little molecular scissors attached right there, ready to snip apart those remaining double sugars. You've got lactase, which breaks lactose into glucose and galactose.
Ah, lactase. We'll come back to that.
Definitely. Then sucrose splits sucrose into fructose and glucose. And maltase and isomaltase handle maltose and other short glucose chains, basically giving us pure glucose in the end.
Okay, so once everything's broken down to those single sugar units, glucose, galactose, and fructose, they need to get inside the enterocytes, those cells lining our gut. This sounds like it probably involves some specific sugar doorways, as you put it earlier.
It absolutely does. Think of them as highly selective bouncers at the cell membrane club. For glucose and galactose, the main player is SGLT1. That stands for sodium galactose cotransporter.
SGLT1, got it.
And this is active transport. It actually uses energy, kind of piggybacking on a sodium gradient, to pull these sugars into the cell, even if the concentration inside is already higher.
Okay, pushing them uphill.
Precisely. Then, to get out of the enterocyte and into the bloodstream, and also for some uptake from the gut, we have GLUT2. That's glucose transporter type 2. This one works by facilitated diffusion, more like an open door when the concentration is right. It handles glucose and fructose.
GLUT2.
Yeah.
And fructose specifically.
Fructose also uses GLUT5 for facilitated diffusion into the enterocyte. The key takeaway here, this is an important anchor, is that different tissues have different types of these GLUT transporters. There are actually GLUT1 through 14, and they have different affinities, different stickiness for glucose.
And one is particularly important for insulin, right?
Yeah.
GLUT4.
Exactly. GLUT4 is found mainly in muscle and adipose tissue, fat tissue. And it's insulin sensitive. Think of insulin as the key that unlocks the door. When insulin is present, it signals the cell to put more GLUT4 transporters onto its surface.
So more doorways open up.
Right, allowing much more glucose to enter those specific cells. This is absolutely vital for understanding blood sugar regulation and conditions like diabetes, where this process goes wrong.
Okay, that makes sense. Now let's tie this whole digestion process directly to a clinical condition many people are familiar with. You mentioned lactase earlier. Let's talk about why some people can't tolerate dairy. That's often due to something called lactose intolerance, right?
Precisely. It's a perfect clinical example of what happens when just one critical enzyme is missing or significantly reduced in activity. The biochemical basis is pretty straightforward. If you have insufficient lactase activity, the disaccharide lactose just isn't hydrolyzed. It isn't split into its monosaccharide components, glucose and galactose, in the small intestine.
And the causes can be genetic, I understand. Like, lactase activity naturally diminishes after childhood in many populations, particularly those of African and Asian descent.
That's very common, yes. It's called lactase non-persistence, but it can also be non-genetic. It can result from injury to the small intestine, maybe due to inflammatory bowel disease like Crohn's or ulcerative colitis or even surgery, or a bad infection that damages the gut lining.
And the consequences, the symptoms people feel, that's all down to what happens to that undigested lactose.
Exactly. Because it wasn't broken down and absorbed in the small intestine, the undigested lactose travels all the way down to the large intestine. Now, the bacteria living there, the colonic bacteria, they see this lactose as an unexpected feast.
Uh-oh.
Yeah, they start to ferment it. This fermentation produces a whole host of organic acids, like lactic acid, remember that name, and various gases like hydrogen, methane, and carbon dioxide.
Okay, so acids and gas. That sounds uncomfortable.
It is. These fermentation products increase the osmotic pressure inside the gut. Basically, they draw water into the colon by osmosis, and that excess water leads directly to diarrhea. Plus, of course, the buildup of gases causes abdominal cramps, bloating, and, well, gas.
So the typical symptoms are things like abdominal pain, general discomfort, bloating, diarrhea, sometimes nausea, usually kicking in maybe 30 minutes to a couple of hours after consuming lactose.
That's the classic picture. And it's really crucial to distinguish this from a milk allergy. Lactose intolerance is not an allergic reaction. The immune system isn't involved at all. It's purely a digestive issue.
Right, important distinction. And diagnosis often involves things like a hydrogen breath test, because those bacteria produce hydrogen gas, or maybe a stool acidity test.
Correct. Those tests can confirm that lactose isn't being properly digested. And management, well, it typically involves avoiding lactose, so a lactose-free diet, or consuming foods where the lactose has already been broken down, like lactase-treated milk, or simply taking lactase enzyme supplements right before eating dairy.
Okay, now this bacterial fermentation in a large intestine, it's producing organic acids, like lactic acid and gases, all in an environment that's pretty limited in oxygen, right? Deep in the gut.
Right, it's anaerobic down there.
This offers a really great parallel, I think. Think about an intensely exercising muscle. Maybe you're sprinting flat out. That muscle also finds itself in a situation where the oxygen supply just can't keep up with the demand. It becomes oxygen limited too.
Absolutely.
So here's a question for you listening to consider. What similar metabolic process might be occurring in your muscles under those strenuous low oxygen conditions? And what common product might be generated there that we also just saw being produced by bacteria in the gut during lactose fermentation? Keep that in mind. We promise we'll circle back to it later.
Good question to ponder.
Definitely. So moving on. It's clear glucose is vital. But how vital? Is it like a universal fuel that every single part of our body can burn equally well? Or are some tissues maybe more, let's say, addicted to it than others?
Glucose truly is the central character, maybe the superstar in our whole energy story. Your body aims to keep blood glucose tightly regulated. Around that 5 milliliter level, it's roughly 1 teaspoon of sugar dissolved in your entire blood volume. And you need around 180 grams of glucose per day overall.
180 grams a day, that's quite a bit.
It is. And you're right, not all tissues are created equal in their glucose dependency. Some have an absolute requirement for it. They need about 40 grams per day collectively. We're talking about tissues like red blood cells, RBCs, white blood cells, WBCs, the inner part of the kidney called the medulla, the testes, and the lens and cornea of the eye.
Why them specifically?
Often it's because they have limited mitochondria or limited blood supply or specific metabolic needs. Their glucose uptake is pretty much directly dependent on how much glucose is circulating in the blood at any given moment.
Okay. And what about the brain? We always hear the brain runs on glucose.
It certainly prefers it. The central nervous system, the brain, has a huge appetite for glucose, requiring about 140 grams per day under normal conditions. It can adapt to use other fuels like ketone bodies during prolonged fasting, but glucose is definitely its primary preferred fuel source.
140 grams just for the brain. Wow.
It's an energy hog. And
Yeah.
beyond that, other tissues like the liver and adipose tissue also need glucose, not just for energy, but for specialized functions too, like synthesizing triglycerides. That's how we store fat for long-term energy reserves. Glucose provides the backbone for that.
Okay, let's just circle back to those red blood cells for a second.
Mm.
You mentioned they have an absolute requirement for glucose, and they're unique because they completely lack mitochondria, right? Those are usually the cell's main powerhouses for making ATP using oxygen.
Correct. No mitochondria in mature red blood cells.
So, given that they need glucose, and they don't have mitochondria to burn it with oxygen, here's our next question for you listening. How do you think RBCs manage to produce the ATP they absolutely need to survive and function, especially considering they can't use oxygen for ATP synthesis? Think about that, and we'll connect this directly to our next big topic.
A very important point for RBC survival.
Definitely. So now we're getting right into the heart of cellular energy production. The glucose has made it through digestion, absorption, it's traveled in the blood, and now it's finally inside the cell. What happens next? Where does the real energy extraction begin?
Here's where the magic truly begins, metabolically speaking. We're talking about glycolysis. The name literally means sugar splitting. This is the central pathway for carbohydrate breakdown for catabolism. And it happens right there in the cytosol, the watery, soupy part of the cell, in essentially all our cells, from brain cells to muscle cells to liver cells.
Glycolysis. Okay, the main event.
It really is. It takes one molecule of glucose, which is a 6-carbon sugar, C-SLOX, and through a series of steps converts it into 2 molecules of pyruvate, each a 3-carbon unit, C3.
One C6 into 2 C3s. And there's an overall equation for glycolysis, right? That might be a good anchor for people to remember.
Absolutely. The net equation captures the inputs and outputs beautifully. Glucose, C6, plus 2 inorganic phosphate pi, plus 2 ADP, plus 2 NAD, plus 2 pyruvate, C3, plus 2 ATP, plus 2 NADH, plus 2 H++, plus 2 H2O.
Okay, quite a bit going on there. Let's break that down.
Let's do it. So it's a sequence of 10 distinct enzyme-catalyzed reactions. And overall, it's an exergonic pathway, fancy term, meaning it results in a net release of energy. Think of it like kicking off a controlled chemical chain reaction that starts to unleash the energy stored within that glucose molecule.
And the key products are?
The key energy products are, first, a net production of 2 ATP molecules per glucose molecule. That's the cell's direct usable energy currency, like getting immediate cash.
2 ATP.
And second, it releases reducing power in the form of 2 NADH plus H plus H plus N. These NADH molecules are like little rechargeable batteries or energy packets. They're electron carriers that will shuttle high-energy electrons off to be used later, primarily in mitochondria if oxygen is present, to generate a much larger amount of ATP.
Ah, so the NADH is like potential energy for later.
Exactly. Now, what truly makes glycolysis unique, really sets it apart, is its ability to operate anaerobically. It can crank out that net 2 ATP even if there's no oxygen around.
That seems crucial.
It is. It's the only major catabolic pathway that can do this. Plus, it's not just about energy. Glycolysis also provides important intermediate molecules, like little building blocks that the cell can siphon off for other essential functions, like making amino acids or fats.
Okay, so it sounds like a really central multi-purpose pathway. You mentioned 10 steps. Can we think about it in maybe broader phases?
Yes, definitely. It's often helpful to conceptualize glycolysis in 2 main phases. You could think of it metaphorically as an investment and payoff scheme for the cell.
Investment and payoff. I like that. Tell me more.
Okay, so phase 1 is the preparative phase, sometimes called the energy investment phase. This covers roughly the first half, reactions 1 through 5, depending on how you count. Here, the cell actually has to spend 2 ATP molecules per glucose to get the process going.
Spending money to make money? Kind of.
Exactly like that, like putting coins into a vending machine before you get your snack. In the very first step, catalyzed by an enzyme called hexokinase, or glucokinase in the liver, an ATP is used to add a phosphate group onto glucose, forming glucose 6-phosphate, G6P. This is a key step because adding that negatively charged phosphate group effectively traps the glucose inside the cell. It can't easily cross back out through the membrane.
Locks it in. Okay, that's 1 ATP spent.
Right. Then a few steps later, another ATP is invested. This happens in reaction 3, catalyzed by a super important enzyme called phosphofructokinase 1, or PFK1. This step forms fructose 1-sulfose 6-bisphosphate, F1-sulfose 6-bisp. This reaction is often called the committing step of glycolysis.
The committing step, why is that?
Because once F16-bis is formed, that molecule is essentially locked into going down the rest of the glycolytic pathway. There's no easy turning back. It's like pushing a boulder past the crest of a hill. This makes PFK1 a major point of regulation, like a crucial metabolic traffic light controlling the flow through glycolysis.
Got it. Investment phase, 2 ATP spent, glucose trapped and committed. Then comes the payoff.
Then comes phase 2, the ADP generating phase or the payoff phase. This covers the remaining reactions, roughly 6 through 10. This is where the cell starts to see a return on its initial investment. It actually generates 4 ATP molecules per original glucose molecule during this phase.
4 ATP generated, but we spent 2.
Exactly. So the net gain is 2 ATP overall per glucose.
Okay, payoff phase nets 2 ATP. How does that happen?
Well, first, that F16 bisphosphate molecule, the one that had energy invested into it, gets split in half. It breaks down into 2 smaller 3-carbon units. One is called glyceraldehyde 3-phosphate, G3P, and the other is dihydroxyacetone phosphate, DHAP, which is quickly converted into another G3P. So from this point on, everything happens twice per original glucose molecule.
Right, 2 parallel tracks now.
Exactly. Now, these 2 G3P molecules go through the rest of the steps, and it's during these later steps that we see the direct payoff. First, in reaction 6, there's a crucial redox reaction oxidation reduction. G3P is oxidized, and in the process, NAD+ is reduced to form NADH plus H plus A. This is where that reducing power, those electron carriers we mentioned, are generated. Remember, this happens twice, so we get 2 NADH per glucose.
Okay, the NADH harvest, then the ATP.
Then the ATP payoff comes through a process called substrate-level phosphorylation.
Substrate-level phosphorylation. That sounds a bit technical. Can you simplify that?
Sure. It's actually quite direct. It means that a high-energy phosphate group is transferred directly from one of the intermediate sugar molecules, the substrate, onto an ADP molecule, instantly forming ATP.
Ah, like a direct handoff of energy.
Exactly. Instead of waiting for the complex machinery in the mitochondria, it's like getting quick cash directly from the reaction itself. This happens twice in the payoff phase. First in reaction 7, creating ATP, and then again in the very final step, reaction 10, catalyzed by another key enzyme called pyruvate kinase, PK. Here, phosphenol pyruvate, PEP, hands off its phosphate to ADP, creating the final ATP and leaving us with pyruvate.
Okay, so 2 steps make ATP directly, and since everything's doubled, that's 4 ATP total in this phase.
Precisely. And that enzyme, pyruvate kinase, PK, brings us to an important clinical link. PK deficiency is an inherited condition. It's autosomal recessive. It particularly affects red blood cells. If RBCs don't have enough functional PK, they can't generate enough ATP through glycolysis.
And we said they rely on glycolysis.
Right. So without enough ATP, these RBCs can't maintain their shape, their membrane integrity, and they end up being destroyed prematurely. This leads to a condition called hemolytic anemia, basically. Anemia due to red blood cell breakdown.
Wow. Okay, so that deficiency directly impacts their energy supply.
It really does.
Yeah.
And this brings us back perfectly to your earlier question about red blood cells. To answer your question for the listeners, because RBCs lack mitochondria, they rely entirely on glycolysis for all their ATP. That direct, quick ATP generation via substrate-level phosphorylation is their absolute lifeline.
Okay, that answers the ATP part.
Yeah.
But what about the NADH they produce in glycolysis? You said that usually goes to the mitochondria, but they don't have any. And glycolysis needs NAD+ to keep going.
Excellent point. They need to regenerate NAD+ from the NADH produced in glycolysis, otherwise the pathway would just stop. They do this via anaerobic glycolysis, converting pyruvate to lactate, which we'll discuss right now. It's their way of recycling the NAD+ out of milk. And there's also another really neat trick RBCs do. An intermediate in glycolysis, 1,4-on-3 bisphosphoglycerate, 1,3-BPG, can be shunted off into a little side pathway to make 2,4,3 bisphosphoglycerate, or 2,3-BPG. This molecule binds to hemoglobin and actually decreases hemoglobin's affinity for oxygen.
Wait, decreases oxygen affinity. Isn't that bad?
Not at all. It means hemoglobin is more willing to release its oxygen to the tissues that need it. So 2,3-BPG helps optimize oxygen delivery throughout the body. Pretty amazing how tightly these metabolic systems are integrated with physiological function.
That is pretty neat. Okay, so you mentioned anaerobic glycolysis and lactate. Let's get into that. What exactly happens when there isn't enough oxygen around, like in that intensely exercising muscle we keep talking about, or in tissues like RBCs that never use oxygen anyway?
Right. This is where anaerobic glycolysis becomes absolutely crucial. It's essentially the cell's plan B for generating ATP when oxygen is limited or completely absent. It's the go-to pathway in cells with few or no mitochondria, like RBCs, in tissues that might be poorly oxygenated temporarily or, very commonly, in vigorously exercising muscles where oxygen demand outstrips supply.
Okay, plan B. So what's the key step? What happens differently?
The key difference happens right at the end. Instead of pyruvate potentially moving into the mitochondria, if oxygen were present, pyruvate stays in the cytosol and gets converted into lactate. This reaction is catalyzed by a cytosolic enzyme called lactate dehydrogenase, LDH.
Pyruvate to lactate via LDH.
Exactly. The simple equation is pyruvate plus NADH plus H plus EO yields aldate plus NAD plus E.
Ah, look, NAD plus is produced.
Precisely. That's the whole point. While the lactate itself isn't immediately used for energy by that specific cell at that moment, this reaction regenerates the vital NAD plus from the NADH that was produced earlier in glycolysis.
So LDH is like the NAD plus recycling plant.
That's a perfect way to think about it. Without enough NAD plus in glycolysis, specifically reaction 6, the redox step, would grind to a halt. LDH keeps the recycler running, allowing glycolysis to continue churning out that small but rapid supply of 2 ATP per glucose, even when oxygen is scarce. It's how your muscles can keep going for that extra burst, or how RBCs survive constantly.
Okay, so anaerobic glycolysis still nets 2 ATP per glucose, but critically, it regenerates NAD+, so the process can continue without oxygen.
You got it.
Now, remember our earlier question about bacterial fermentation in the large intestine during lactose intolerance? We asked what similar product is generated compared to an exercising muscle. The answer is
Lactate.
lactate.
Yeah.
Just like your muscles produce lactate under anaerobic conditions, low oxygen, the bacteria down in your gut, also in an anaerobic environment, ferment that undigested lactose and produce organic acids, including lactic acid. That's what leads to those unpleasant digestive symptoms. See the parallel?
It's a great example of convergent biochemistry in very different settings. And here's where it gets really, really interesting. What happens to all that lactate?
Yeah, is it just a waste product then? Or did the body do something with it?
Far from being just waste, lactate is actually a really important mobile energy currency. Once it's produced, say in muscle or RBCs, it's released into the bloodstream. From there, it's transported to other organs, particularly the liver, the heart, and the kidneys, which can actually use it.
Use it how?
Well, your heart muscle, for instance, is very good at taking up lactate from the blood, converting it back to pyruvate using its own LDH enzyme, and then completely oxidizing that pyruvate in its mitochondria to generate lots of ATP. The heart actually quite likes lactate as a fuel.
Wow. Okay, so the heart can burn it. What about the liver?
The liver, and to some extent the kidneys, are incredibly clever. They take up lactate from the blood, convert it back to pyruvate, and then, using a pathway called gluconeogenesis, they essentially run glycolysis in reverse to convert that pyruvate back into fresh glucose.
Wait, they turn lactate back into glucose?
Yes. This newly made glucose is then released back into the bloodstream, where it can travel back to the tissues that need it in the first place, like the muscles or RBCs, to be used again. It's a beautiful recycling system.
This whole cycling of lactate from tissues like muscle to the liver to be made back into glucose, and then the glucose going back to the muscle, that sounds like a loop. Does it have a name?
It certainly does. This metabolic partnership between peripheral tissues and the liver is known as the Cori cycle, sometimes called the lactic acid cycle. It's a really crucial mechanism for regenerating glucose and allowing tissues to continue functioning under anaerobic or low oxygen conditions. It's like a lactate shuttle and glucose regeneration loop.
The Cori cycle. Okay, another important concept. So lactate isn't waste, it's fuel and a precursor for making new glucose.
Exactly. And lactate is actually produced continuously, even when you're resting. Your body makes about 40-50 grams of lactate per day, just from normal metabolism and RBCs, skin, brain, skeletal muscle, even the GI tract.
Really? Even without exercise?
Yeah. But during strenuous exercise, production skyrockets. Plasma lactate levels can easily increase tenfold in just a few minutes. You might produce 30 grams in just 5 minutes of intense activity.
Wow. And it also increases in illness, right? Like low oxygen states.
Yes, pathological situations involving hypoxia, meaning low oxygen delivery to tissues, also lead to significantly increased lactate production. Things like shock, severe sepsis, congestive heart failure, or blocked arteries can all cause lactate levels to rise.
So the concentration of lactate in our blood seems to be this really delicate balance then. between how much is being produced, how much is being used by tissues like the heart, and how much is being cleared or recycled by the liver and kidneys.
Precisely. It's a dynamic equilibrium.
And when that balance gets seriously disturbed, that's when problems arise.
That's when we can see clinical states of elevated lactate. Mild elevations, say between 2 and 5 millilem, are often termed hyperlactatemia. At this level, it's still below the point where the kidneys start spilling it into the urine, and importantly, your blood pH is usually maintained pretty well because your body's buffering systems can cope. You might see this after very strenuous exercise, or sometimes even after a large carbohydrate-rich meal.
Okay, hyperlactatemia, usually manageable, but what if it goes higher?
If plasma lactate climbs above 5 millilem, that's when we enter the territory of lactic acidosis. Now, the lactate level is above the renal threshold, meaning the kidneys can't reabsorb it all, and critically, the body's buffering capacity gets overwhelmed, leading to a detectable drop in blood pH. This acidosis is a serious, often life-threatening clinical condition that requires urgent medical attention to figure out the underlying cause and correct it.
And while we often instinctively link high lactate and acidosis with the lack of oxygen hypoxia, you're saying it's not always that simple.
Exactly. While hypoxia is a major cause, lactic acidosis can definitely occur even when oxygen delivery seems perfectly normal. There might be underlying problems with how lactate itself is processed.
Like what?
Well, severe liver disease can impair the liver's ability to clear lactate via the Cori cycle. Kidney disease can impair its excretion. Certain vitamin deficiencies, like thiamine, vitamin B1, which is crucial for pyruvate metabolism, can cause lactate to back up. High alcohol intake can mess with the liver's redox state and impair gluconeogenesis from lactate. Inherited deficiencies in enzymes involved in pyruvate metabolism or even certain drugs and toxins can also lead to lactic acidosis. It's a complex diagnostic puzzle sometimes.
It definitely sounds like it.
Wow.
Okay, that was a truly insightful deep dive into carbohydrates and glycolysis, touching on everything from basic structures to clinical conditions. So just to bring it all together for our listener, especially thinking about that first-year medical student perspective, what are the absolute big takeaways about glycolysis? What's the essential knowledge they need to anchor?
Okay, the big picture for glycolysis. First, it's absolutely central to carbohydrate breakdown to catabolism. It happens in the cytosol of basically all tissues.
Central, cytosol, all tissues.
Got it. Second, its core function is to oxidize one glucose C6 molecule into two pyruvate C3 molecules.
One C6 to two C3s. Check.
Third, the key energy outputs are a net production of 2 ATP via substrate-level phosphorylation for immediate cellular energy, and 2 NADH plus H-platypeptide, which represent reducing power potential energy for much more ATP later if oxygen is available.
Net 2 ATP, 2 NADH.
Fourth, its unique and critical ability is that it can operate anaerobically without oxygen, making it vital for RBCs and exercising muscle. It's the only major pathway that does this.
Anaerobic capability, crucial.
Fifth, it's not just about energy. It also produces vital intermediate molecules that can be used as building blocks for other biosynthetic pathways, like making amino acids or lipids. It feeds into other cellular factories.
Provides intermediates.
And finally, when oxygen is available, the pyruvate produced by glycolysis serves as the crucial feeder molecule for the next major stage of energy extraction, the TCA cycle, or Krebs cycle, inside the mitochondria. Glycolysis sets the stage for aerobic respiration.
Feeds into the aerobic pathway. Excellent summary. What an incredible journey, really, from the simple structure of a sugar molecule all the way to how our bodies make this vital energy, even managing without oxygen when necessary. It's so clear that this is just a fundamental process, a real metabolic bedrock.
Indeed. And understanding these stages of catabolism, especially glycolysis, it's like having a foundational map to the body's entire energy landscape, isn't it? It helps explain so much from, why we feel that muscle burn during an intense workout, that's the lactate buildup, to why certain metabolic conditions like PK deficiency or lactose intolerance manifest the way they do.
And the interconnectedness is just amazing how a seemingly simple dietary issue like lactose intolerance can lead to the production of the very same metabolic product, lactate, as an intensely exercising muscle, all linked by that clever need to recycle NAD+.
It really highlights the elegance and efficiency of metabolic pathways, and it naturally raises an important follow-up question for you, the listener, to ponder. We focused almost entirely today on breaking down carbohydrates for energy catabolism. But what happens when the body has excess glucose and needs to build up energy stores or synthesize complex carbohydrates? How does it switch gears from breaking down to building up or storing glucose as glycogen? And what role do hormones, like insulin perhaps, play in orchestrating this intricate dance of energy management between catabolism and anabolism?
A great question for further thought. How does the body manage the flip side, building and storing? Something to definitely mull over as you continue your own deep dive into biochemistry.
Keep asking those questions.
Absolutely. Well, thank you so much for joining us on the deep dive today. Keep exploring, keep questioning, and we'll be here to help you unpack more fascinating topics in metabolism and beyond. Until next time.